Smear for gram staining purpose determine and confirms the presence of bacteria and yeast to establish the presence of potential pathogenic organisms. Gram stain is essential in evaluation of all suspected cases of bacterial meningitis. 3. Gram staining smear are also scanned for the presence or absence of pus cells indicative of infection and squamous epithelial cells indicative of mucosal contamination. 4. A sputum specimen showing more than 25 squamous epithelial cells per low power field regardless of the number of pus cells indicates that the specimen is grossly contaminated with saliva and that the bacterial culture should not be performed. Principle: to demonstrate the differential staining properties of bacteria, gram positive bacteria retains the gram crystal violet dye after decolorization and appears blue or purple blue. Gram negative bacteria are not capable of retaining the crystal violet dye after decolorization and are counterstained red by dilute carbonyl function. Performance of specimen: the organism isolation and identification will be usually performed only if culture is requested. Primary samples: urine, sputum, throat swab aspirated from different sites of catheter, swabs from different sites, pus, CSF and other body fluids. Tissue: bone marrow, biopsy materials, endometrium and surgical tissue, semen. Type of containers and additives. Urine: instruct the patient to collect clean catch midstream urine specimen in a sterile wide mouth plastic container. Sputum: instruct the patient to collect a deeply coughed expectorated sputum in a sterile wide mouth container after rinsing the mouth with water. Throat swab: collect the specimen in a sterile cotton swab, swabbing the tonsillar fossa and oropharynx. Use a sterile tongue depressor if necessary. Aspirate from different sites and catheter. Insert to collect the specimen aseptically in a sterile screw capped container. Swabs from different sites: collect the eye swab, congenital swab specimen, swabbing the lower inferior dorsal conjunctival fornix, avoiding the eyelid border and lashes with a sterile cotton swab. Collect the ear swab specimen, swabbing the external ear gently, avoiding the pinnae with a sterile cotton swab. Collect the nasal swab, inserting the sterile cotton swab gently through the nostril or to the nasopharynx, swabbing the anterior nair with a sterile cotton swab. Collect the wound swab specimen, swabbing the wound site gently with a sterile cotton swab. Collect the material from the posterior fornix of vagina, swabbing that area with a sterile cotton swab. Collect the male urethral discharge with a sterile cotton swab. Instructed to collect a high vaginal cervical swab, avoiding the contamination in a sterile cotton swab, and transport the specimen to the laboratory within 1 hour of collections. Do not accept dry swab specimen from other labs or hospitals. First, instruct to collect the specimen in a sterile screw cap test tube or to send in aspirated syringe, CSF and other body fluid. Instruct to collect the specimen in a sterile screw capped test tube. Tissue: bone marrow, biopsy materials, endometrium and surgical tissue. Instruct to collect the specimen in a sterile screw capped test tube containing 5 ml sterile normal saline specimen. Instruct the patient to collect in a sterile wide mouth container. Reagents: microslides, sterile normal saline, diamond marker, tissue paper, gram stain, immersion oil. Equipments: autoclave, biosafety cabinet, microscope. Procedure: make a smear of specimen on a clean labeled microslide and allow to dry. Heat fix the smear to the slide by passing the slide 3 to 4 times through the flame of a Bunsen burner so that the smear does not wash off during the staining procedure. Gram staining: 1. Stain the slide by gram staining procedure. 2. Cover the smear with crystal violet and leave it for 1 minute. 3. Wash the slide with water. 4. Cover the slide with the gram saline and leave it for 1 minute. 5. Wash the slide with water. 6. Decolorize quickly with acetone, rocking the slide gently. 7. Wash with water immediately. 8. Counterstain with a dilute carbonyl fusion for 30 seconds. 9. Wash with water and blow dry. Observe the Observe the stain smear under the oil immersion objectives for gram-positive bacteria that appears blue or purple-blue and gram-negative bacteria that appears red or pink, and for cells. Interpretation of results. Report the results as follows. 1. For sputum samples, assesses its quality by Bartlett scorecard given below. Grade the report vaginal smear as described by the Nugent et al. Given below. Mention the number of cells per oil immersion field for epithelial cells and pus cells. 4th, indicate the morphology and arrangement specifically of predominant bacteria and either gram-positive cocci or gram-negative bacilli. 5th, mention the Coca bacillary or other forms if seen. Number of neutrophil per 10x low power field and grade less than 10 grade 0, 10 to 25 grade plus 1, more than 25 grade plus 2. Presence of mucus plus 1. Number of epithelial cells per 10x low power field and grade 10 to 25 grade minus 1, more than 25 minus 2. Nugent scoring of vaginal swab for diagnosis of bacterial vaginosis. Lactobacillus species.